Okay, so today, new unit. This will be a longer unit. It's kind of similar to the genetic algorithms unit. Some of this will be review for many of you who have some background in machine learning, but I'm still hoping that this will uncover a few dimensions that maybe you haven't gotten, because we kind of go back to a little bit of the fundamentals. For those of you who haven't worked directly with neural network approaches in machine learning, then hopefully this will be kind of a useful introduction to that. So, like I said, we're starting on neural networks, and so this is a bio-inspired course, and so I want to kind of talk about where these things come from. And so neural networks are inspired by the the neuron, which is just a special type of cell. And so if you think about eukaryotic cells, you've got a cell body that I can kind of draw sort of in the center here. And in the case of neurons, this cell body has these things coming off of it that kind of look like branches of a tree. Like if you were to look up at a large tree, you'd have multiple branches, and each one of it would have multiple branches. And those are things called dendrites. And so I might simplify that drawing by just showing kind of, maybe something that looks along the lines of this. Something along this lines. I'm going to leave this open for a second. And these are more sort of branch-like. And so these are called dendrites. Well, so the cell body goes by the name of a soma. And attached to the cell body are these things called dendrites. And because they kind of look like tree branches, if you were to sort of stand this thing up on end, sometimes people call this like a dendritic arbor. Because it's kind of tree-like. And so this cell then also has a special part attached to it, this kind of long part. And if I really want to be honest about it, it kind of has a bunch of these little nodes. Didn't mean to erase the whole thing there. So a bunch of these little kind of nodes here. And then that ends up terminating in, I'll draw this as another type of, a little bit of a branching structure, but not quite as much as the dendrites. And this thing here is sometimes called a nerve fiber, or as you might be more familiar with, an axon. And so you have axons and dendrites. And so this is, there are many different types of neuron. This is the most common type, and this is what I'm referring to as a multipolar neuron. And all that means is that it's got one axon and many dendrites. Okay, and so at the end of this, at this sort of axon end here, then what you're going to find is there's going to be other neurons that this axon terminal here, this kind of nerve ending over here, will end up interfacing with other dendrites of other neurons. And so drawing one neuron here, but you could then imagine that there would be on this side, another neuron's dendrites. And so this would go off. And so this space here, so this is So these are the dendrites of another neuron. And this space, maybe I'll highlight it, between axon and dendrite, we refer to as a synapse. And that is the space between axon end and dendrite. And it's just a gap. And you can actually have self loops. So you can actually have, I've drawn this kind of straight, but this long axon here, this long nerve fiber, can actually bend around and interface with its own synapses. And those are often called autopses. So auto means self. And so a self synapse, they just call an autops. And so you can have those sort of self loops. But the kind of simplest version here is every neuron connects to a bunch of other neurons. So what do I mean by a bunch? So as an example, in the human brain, so the central nervous system, there are about on the order of 10 to 11 neurons, a lot of neurons. And by the way, these are cells. So you develop them. So most other cells in your body go through mitosis. They grow and they die. Not so much your neurons. Your neurons, there are stem cells initially that turn into neurons. You get most of your neurons during your development. You keep them for your whole life. Once they die, that neuron is kind of gone. Neurons don't reproduce. You do keep around more. You can get neurons added later in life, even up as late into your upper 20s. You could still have these, even later than that, but to a much lower extent. So most of this neuronal growth goes on during early development. Now, you can get modifications to these dendrites can occur on life history scales. And so you can get more dendrites, you can have modifications to how the dendrites work, you can get less dendrites. So you can have this so-called synaptic pruning that can happen later. So there's a lot of modifications that go on to each individual cell, but you don't get more of these cells. You're kind of stuck with your brain once it's kind of grown up. Now, you got all these neurons, so how many connections do you get? Each neuron, so in the human brain, each can have as many as 10 to the third or 10 to the fourth connections to other neurons. So I've drawn a very simple looking picture here where it looks like one neuron maybe has the opportunity to interface. So this neuron interfaces with this neuron, maybe interfaces with a couple of other neurons, but these dendritic arbors are so complex that they can actually fan out and connect with, 1,000 other neurons. And so you can have one neuron interfacing with a huge number of other neurons. And that goes on for each one of these. So it's an extremely complex network in your brain of these neurons connecting to a bunch of these other neurons. And that's just sort of the topology of it. And like I mentioned, axons can connect to your own cell body. That's so-called autops. And so we really focus on what happens at this junction, this synapse. And I'm going to describe that in just a second. But like I said, these properties that I described can change over time. They can change possibly even over a class period. And so I'm going to get into that. So what goes on with neurons? So the idea here, since our kind of basic picture, is that there are signals from other neurons that are received. So I guess I can maybe draw the directionality here. So this is kind of the direction of information flow, traditional information flow down a neuron. Signals enter here. So this is where signals enter and are summed. And then when we get what I'll refer to in a second here, and I'll describe this as an action potential, then you can generate a new signal. And so this is sort of the output over here. And so the output transmits that signal through this synapse. And when we're focusing on a synapse, we sometimes refer to the neuron that is upstream as being presynaptic. I don't know if I can fit that, presynaptic, and the neuron that is downstream as being postsynaptic. It's just kind of lingo there. So that's kind of the information flow. So on this page, it's flowing from left to right. And so, like I said, just in the normal cells. And so every cell in your body establishes what's called a membrane potential. So it just so happens that there are ions that exist inside every cell of your body that, due to actually things related to Boltzmann, so due to diffusion and things like that, then they're just naturally, you can get positive charges inside the cell will start diffusing out of the cell and creating a charge imbalance. So what happens is the insides of your cells, all of your cells, get negative, and the outsides of your cells get a little more positive. Now, once you get enough positive charges going over here, then this natural diffusion process starts to get turned off because of the charge interaction. So once you get too many positives out here, then there's kind of an energetic balance where things just kind of come into rest, where every cell of your body rests at like minus something like 70 millivolts or something like that. So if you were to take a little voltmeter and actually measure from inside of the cell to the outside of the cell, you would see a little voltage drop of around like minus 70 millivolts. So just every cell sits there. Neurons are special types of cells where the inner workings of the cells are specially tuned to these voltage changes so that if you can change the inner voltage a certain amount, it will generate a voltage spike that we refer to as an action potential that will travel down the body of the neuron and will end up interacting with the voltages inside this neuron over here. Now, in the movies, in TV shows, they often like zoom in on like people's brains as they're thinking, and you see these cartoons with like sparks, and all of these things lighting up and all that. None of that happens. So there's no actual sparks or anything like that. It's a lot kind of less exciting. Most of the synapses that you have are these chemical synapses that happen over here, which I'll describe here in a second. There are electrical synapses where ions can flow directly from one neuron to another at a number of other places, but they're very rare. And occasionally you do get electromagnetic coupling between neurons where you don't actually have to have material flowing from neuron to neuron, but one neuron sitting next to another neuron can kind of make it more or less prone to fire. And that's something called ephaptic coupling. And that's also very rare. So most of the signaling between neurons is chemicals that are shuttled from one neuron to another. And in general, let me go to this page here. In general, when we refer to chemical signals between cells, we call those things hormones. So we talked about ants right now, so I talked about ant colony optimization. They laid down a chemical signal in the environment, and then that ended up causing changes in behavior in another ant. So when you have chemicals communicated among individuals of the same species, then you refer to those as pheromones. When you have chemicals in between cells inside one individual, we call those hormones. Now there's a special type of hormone called a neurotransmitter. And a neurotransmitter are the chemicals used for communication between neurons. Okay, so what happens here? So if we look at a synapse, so I focus on a synapse here. So we've got some, so we had some axon here. So this is our axon. And then we've got some dendrites here. So maybe I'll draw this a little bit bigger. So this is kind of a dendrite coming off of some ugly. So this is my dendrites. And then the synapse is in between here. So there's a dendrite. All right, so what happens when a synapse fires is there is a release of neurotransmitters. So when a synapse fires, then you get a release of neurotransmitters from an axon into the gap, into the synaptic gap, synaptic cleft. So, so I'll say into the synaptic, and I'll just call it a gap here. And so you get this release of these neurotransmitters, and they're not charged particles or anything like that. There's nothing electrical that's going on here. These things just get released into this gap, and they float from one spot to the other. So there are mechanical properties at play here that govern how quickly these signaling molecules can get from the axon to the dendrite. And so it ends up being a space where your brain gets to modulate the activity of your neurons. So if you take a certain pharmaceutical or whatever, a lot of times those pharmaceuticals, they actually change the chemical properties of what's going on in here to make it easier or harder for neurotransmitters to get from one spot to the other. And that's what, if you have somebody who has bipolar disorder and so on, these sorts of things can make these synapses, it can mute them or amplify them. And it's just by tuning the kind of statistical mechanical properties of what goes on in this gap. And so these chemicals are released, and they then are going to be received by these receptors on the dendrites. So there's receptors. And when they're received by these receptors, then we get what is the generation of something called a synaptic potential. So the receptors generate a small synaptic potential. And remember, I said the inside of all cells has negative voltage, as it sits at a negative potential. And when you get a receptor here, it's going to use energy inside the cell to actually change the distribution of charged particles inside the cell for a small period of time, so that instead of this cell sitting at whatever minus 70 millivolts, in this area, it might jump up to minus 60 millivolts. So if you were to look over time, the cell in this area might be normally sitting at like minus 70 millivolts, but right after these chemicals are hit by these receptors, then they'll bounce up for a short amount of time and then come back down. And that synaptic potential, that is this little bump in voltage. Now, remember, there's a lot of dendrites, and they're connected to a lot of neurons. Also, the process that shuttles these by, this can be kind of bursty. So you can get a burst of neurotransmitters, and then a small fraction of a second later, another burst over there. And so if you've got synapses that are receiving enough neurotransmitters, or one synapse that's getting hammered, then this little synaptic potential starts to become much larger. So these synaptic potentials add. And eventually they add to the point where they're going to hit a threshold that's going to generate this thing called an action potential, which I'll describe here in a second. So, so we get additive effects from short sequential hits from one presynaptic neuron, or the combination of multiple neurons firing at roughly the same time. And both of those things can cause these synaptic potentials to sum. So these can sum up to polarize, or so sorry, to depolarize the cell. So remember, every cell in your body sits at like negative whatever 70 millivolts. It's polarized. Every cell of your body has got a little bit of a little bit of negative potential. And so what this is doing is this is causing the cells to get rid of that polarity, because they're adding a little bit of positive potential. And so they're gradually depolarizing the cells. They're bringing them closer to 0 millivolts, or even to go higher into positive. So that's what goes on with a synapse. You're generating small perturbations in the cell's voltage, and eventually you'll reach a threshold amount so that inside the cell, so if I go back to, if I draw my kind of cheap and dirty neuron, so this is my synapses, here's my axon, so this is my, so here's my axon, here, and these are my dendrites. And so with accumulation of enough synaptic potentials, the cell body reaches a threshold positive voltage. And this triggers an action potential. And so if I were to draw an action potential, I could do it up here on time. Maybe I'll draw the axes a little different here. We've got, again, the cell is normally sitting down here at whatever, minus 70 millivolts. So, so it's sitting around there resting. This is our resting potential. And then at some point, you end up getting an accumulation of these positive charges, and there ends up being this threshold voltage up here, this threshold. Where if you can manage to get over that threshold voltage, then what's going to happen is it suddenly starts a positive feedback process inside the cell, where the cell starts reinforcing that positive voltage even more all by itself. So this here was driven by synaptic potential. So synaptic potentials. But at this point here, you trigger that action potential. And what that means is the cell is going to start further polarizing or further depolarizing itself, making itself more and more positive voltage as much as it can. And it goes up to a terminal point, which doesn't depend on how strong the signal is. So every cell goes up to this point, and then it kind of flattens out, and then it will come back down, and it goes below its resting potential, and then it comes back to rest. And so this whole thing again is called the action potential. And this period over here is called the refractory period. And so the whole point of this is that you can get a stimulus. So you get a stimulus trigger, and that will generate this pulse that, and we'll talk about what this pulse does here in a second. And that pulse will stay on for a certain amount of time, and then it will turn itself off. And for the refractory period, it will be very hard for any further stimulus to turn it on. So in other words, in order to generate another action potential, the stimulus that's present has to stay there for a long amount of time, because the same stimulus that triggered the action potential here won't trigger it during the refractory period. So it kind of becomes numb during the refractory period. And so, so based on how strong the stimulus is, you get either one action potential or maybe multiple action potentials, but they tend to spike. So you tend to have an on period and then an off period. Now, this is all happening inside the cell body. So that's going to happen. And that action potential this whole thing will end up happening right here at this place at the beginning of the axon, where it's called the axon hillock. It's kind of like the end of the cell body. And that action potential will propagate down the axon. And so basically you're going to get an action potential here. And that action potential is going to end up depolarizing the section of the axon next to it, causing it to have its own action potential. And so then you end up getting this thing moves over, and this thing moves over, and this thing moves over. And so you get the propagation of the action potential. Until it reaches the end of the nerve fiber, the end of the axon here, in which case then it releases neurotransmitters. So then after all of that, you get a release of neurotransmitters. And the process stops. Or I mean, it might be restarted, but that one action potential is done. Hits the end, it releases that. Now again, those neurotransmitters, they're not charged particles or anything like that. They're just chemicals, they're just hormones. And those get released into a gap, a synaptic cleft, a synaptic gap, and that can start the process, or it can at least tickle one other neuron after it. So the idea here is we get the summing action of a bunch of neurons in the body of one neuron. If you get enough, if this neuron is tickled enough by either one really loud neuron or a bunch of neurons firing at the same time from their axons, that will generate a stimulus that causes a potential to go down this whole area here, causing it to release its own neurotransmitters. And what's cool about this whole process is the sensitivity of the synaptic potential can change over time. In other words, if I get a neurotransmitter, like after this neuron fires because it receives some neurotransmitters, it can actually build more receptors on the ends of its synapse. So it can then become more sensitive to that neurotransmitter, so it needs less of a signal in the future. And that is what we call learning. That's what we call neuroplasticity. So neuroplasticity is when you have changes, so persistent changes in the sensitivity of neurons to their stimuli. And so what we'll learn about when we talk about spike time dependent plasticity is there are mechanisms that are thought to exist in the brain that, let's say, every time two things, every time, a bell is rung, you smell food, kind of Pavlov's dog. Well, those are two different inputs, but they coincide in time. If you do that over and over and over again, then both of those stimuli over time will become, your sensitivity to to both of those stimuli may actually increase. And so if you already are sensitized, so when you smell food, you're like, oh, processes go in your brain. I smell food, I start getting hungry. If you always fire when you smell food, and you just so happen to also hear a bell the same time you smell food, then the synapses that got tickled by that bell that weren't enough to cause you to fire, cause that action potential to fire, because another thing was strong enough to make them fire. The neuron doesn't know the difference between this stimulus and this stimulus, and it ends up reinforcing both of them. And so over time, if that bell always happens when the odor of food comes in, you become super sensitive to the bell as well. So that eventually you don't even have to smell the food, you just ring the bell and your mouth starts watering. And that's an example of neuroplasticity, where your individual neurons become more sensitive to at certain synapses than others. So for certain neurotransmitters than others. And that is how we think that the brain learns, is that over time it reinforces certain synapses and it actually can inhibit others. So that's kind of the other thing that I've mentioned here, is that neurotransmitters, or that I haven't mentioned so yet, but neurotransmitters can be excitatory. So this is like positive, or inhibitory. And that's like negative. Maybe I'll go and move that positive up to here. And so, this adds a little bit of texture here. You can actually have an action potential causing a synapse to fire, and that neurotransmitter desensitizing the next neuron. So you can now have neurons that make downstream neurons, postsynaptic neurons, less sensitive, or you can make them more sensitive. So in other words, you can have synaptic potentials that are positive or synaptic potentials that are negative. And when they are finally received in the cell body downstream, postsynaptic, all of them get summed. The positives move you closer to the action potential, the negatives move you farther away from the action potential. So it's a way of summing all of these inputs, some inputs positive and some inputs negative. And that's how neurons communicate with each other. So if we go back to this basic picture of the neuron, there are a lot of people focus on the kind of electrochemical network, and I think they overplay the kind of electrical part of this. It is true that inside the cell body, there is internal communication, which is by voltage gradients. And so you end up getting high voltages here, which become high voltages here, which become high voltages here, and so on. And that's how you get down the cell, from the cell body through the nerve fiber to the end of the axon. But in between neurons, almost all of your synapses are chemical. And those chemicals aren't like ions or anything like that. They're just hormones being dumped into a gap and then being received. And the neurons are adjusting how sensitive they are to those hormones, or how sensitive they are to putting them out into the synapse. There are, I guess, a couple of electrical type synapses that operate a little bit differently, but they're in the minority. Most of the time, people talk about synapses, they're talking about this, which is the chemical synapse. And that's what the artificial neuron is based on, this picture. And so we're going to draw the artificial neuron, which is in every feed-forward neural network that that Google uses out there. Then eventually, in a couple of days, we'll talk about spiking neural networks, which is kind of in neuromorphic engineering, where they actually take this a little bit more seriously, where you actually have built artificial networks, artificial neurons that spike. And what are the advantages of those? We'll get to that. But for most of the neural networks that you hear about in modern machine learning, modern deep learning, are going to be modeled after this chemical communication between neurons. Okay, so before we get into the artificial neuron, I want to stop there to say, are there questions about this? By the way, the way they figured all this stuff out is by studying, and they figured all this stuff out like 100 years ago at this point. And it was by studying giant neurons. And so there's like a squid, for example, that has a neuron that you can like lay out on a table. And that macroscopic model allowed them to actually take, go back to your first circuits class or something like that you might have had to take in the kind of engineering requirement, and allowed you to actually measure voltages down this giant body. And they could then figure out how this stuff worked. Nowadays, we have more methods where we can start, doing this in kind of smaller scale, but still a lot of the models that we have are based off of these model systems with giant neurons, where we just pick out that giant neuron with kind of this macroscopic model that we kind of assume is what's going on at the small scale as well. All right. So any questions about this stuff, about the basic, yeah. I would say the latter. So I would say if I were we have positive synaptic potentials, which if this is resting, they go up and then come back down. And these tend to depolarize. And then we have negative synaptic potentials. And so this is resting potential. So if I've got my resting potential, they go the other way. And they tend to polarize. And so these things can sum. Yeah.
Regarding the refractorization period.
Refractory period, yes.
How long will it take? And second question, based on the artificial neural network, how will that does it play out?
Okay, great. So the first question is, how long is the refractory period? It's going to vary on the type of neuron, but we're talking about fractions of a second. I mean, so this whole, one synapse is, I don't know, you'd have to actually talk to an electrophysiologist here, but it's like hundredths of a second, thousandths of a second, and the refractory period is a fraction of that. You can get several hundred action potentials in a second, so you're going to get bursts of them. You can measure them. I mean, so you can actually you can there are these methods where you actually can record the voltages. So you can basically put a glass probe on a neuron and record voltages as they go by here. And so you can count the number of spikes you get. All the spikes are the same height. So again, the strength of the signals is not encoded in the height of the spike, it's in the frequency of the spikes. So a really strong signal will generate a lot of action potentials right next to each other, and you get this like, it's like a burst. But then a really weak signal gives you like a boop, boop, and there is spontaneous activity as well. So if you record from your brain, you're constantly getting these little blips. But then if like an odor comes in, a perfume or whatever, then certain neurons will just get this like where we're like, oh, you just detected that perfume. And so the width and the density of that really encodes the strength of the experience. Now that gets to your second question, which will go into what I'm saying here. In an artificial neural network, that the traditional artificial neural network, not the spiking ones we'll talk about in a couple of days. In the traditional artificial neural network, there isn't any spiking. We sort of turn the spike strength, we almost turn the things into rates. And so like I said, the strength of an action potential is the number of spikes you get in a row. But the strength of the equivalent in an artificial neural network is going to be a number that's higher or lower. So these action potentials, their heights are fixed, but their frequency and time is not. In a neural network that you would go into TensorFlow and build or something like that, or PyTorch or whatever, those neural networks that you build, they aren't spiking, and so then everything's had a value. So there, it's almost as if the height is what encodes the value. we'll get to all of that too, the activation there. So yeah, well, let's pause on that for a second. That's kind of like my next slide there. So any other questions? I see a question online. So there's a question about habituation. So is habituation when the receptors on a neuron are blocked or the cell changes over time to have less receptors? So habituation is a process where you learn to ignore a signal, and that is a much more complicated learning process. So so learning itself doesn't involve just one neuron. It involves multiple neurons, a system of neurons, and you typically, when you habituate to something, you are still responding to it. So it's coming in through your senses, and it is generating a signal, but the downstream processing is learning to not generate an action due to that signal. And there are a lot of really cool things we could talk about that are kind of a little bit outside of the scope on things like this thing called latent inhibition, where you can train an animal or a neural network to ignore something because it is not rewarded. And then you can start rewarding that thing, and you can see how quickly will it learn to start paying attention again. And you find that certain animals will very quickly learn to pay attention to something that they'd just previously ignored, where other animals will take a really long time to stop ignoring. And you can characterize an individual's latent inhibition, and it usually correlates with conditions like ADHD. So I would never say that ADHD is related to a single neuron. It's a system of how things are wired in the brain. But so I'm just trying to say it's a much more complicated question, but it's a very interesting one, is in what's going on during habituation. And habituation is a type of learning process. But again, that's a little bit out of scope, but I hope at least that kind of heads in the right direction. One other question. So the greater the frequency of signals hitting a dendrite, the greater the propensity to trigger the neuron. That's correct. And the signal down through the axon downstream, that's correct. And the weighted sum of different frequency signals combine to either trigger or not trigger the neuron. That's correct. And that's what we're going to capture in our artificial neuron, which is the basis of pretty much every neural network that you hear about, except for these kind of more exotic neuromorphic cases. All right, so any other, one other thing that popped up there online. Okay, good. Any other, question?
So this model, this seems like a very look at it as like a graph, things, there's cycles, like you have certain dendrites like going to the same axon,
right?
And you're calculating things at like different time steps, and like it's like continuous and ongoing.
That's right.
Whereas like all the neural networks I've seen, it's all like directly cyclic graph. You calculate things kind of like one after the other at the same time step.
Right.
Are there any models? Like, are there any ways of doing learning or optimization that like are closer to this, where it's more ongoing and more like a graph than like the traditional neural networks?
Right. So to summarize that, so the question I think was, that if we look at the kind of natural neurons here, they're asynchronous, they're recurrent, so they have a lot of self loops, they're fully connected. It's a mess. And most of the neural network models that you might hear about, like the ones I'm just about to start talking about, are much more synchronous. They may be very often are feed forward. And we'll talk about what that means here in a second. There are, and we will introduce some of these. Recurrent neural networks are one example where we start adding loops and we see how that works. And then when we get to neuromorphic engineering, where we actually start building devices that spike, things are asynchronous at that point. So there's no notion of a clock or a time step with neuromorphic circuits, where we actually have spiking neural networks, where we really just allow the things to go, and the spikes kind of generate their own events, as opposed to waiting for the next clock cycle to do a computation based on the previous one. So we'll get to that. Yeah. So there's a question about, so if you did use a spiking, like a neuromorphic model, would it be more efficient or accurate? What would be the advantages? And we'll get to that when we get to, so we start talking about neuromorphics. But the long story short is one of the nice things about neuromorphics is that they're only on for the periods of that they're spiking. So they do tend to use less energy. At the moment, they're a little harder to train, but people are figuring out how to do that. They don't scale quite as well in terms of the devices we have, but people are working on that. So they have a lot of promise to provide us a lower energy way of providing sort of small scale neural networks that have lots of capabilities. And that's why people are kind of interested in neuromorphics. Plus, I mean, there are people interested in neuromorphics because they're just more similar to natural computation, and we're always, amazed at what nature can do, and we're not really amazed at what Google can do. I mean, no offense, Google, but I mean, it's, but Google doesn't do what the human brain does, right? And so how do you get there? And some people think, well, maybe the gap that we need to cross, the synaptic gap here, is that we're leaving out so many details of these neurons, so many degrees of freedom that we haven't exploited. And if we make them more biophysical, then maybe we'll close that gap. So that's kind of the two reasons to do it. It's more like the systems we want to mimic, and it's clearly more energy efficient. Okay. All right, so let's move on here. So that's the biology, the kind of neurobiology. So that inspired the basic model of a neuron. And I mentioned last time that a neuron is really no more than a type of nonlinear regression. And I'll kind of show you what I mean by that here. So in a neuron, we assume we have certain features of an input space that we're interested in. And I'll give concrete examples here in a second where I'll actually draw this out, but you can think of a feature might be, if I'm analyzing I'll give a car example later, and it might be the weight of a car, and it might be how fuel efficient it is, or there'd be things you can measure. It might be the color of a pixel. It might be where a pixel's located. These are things you can measure about the outside world. So these are things that come from a sensory system. So we have a set of features, which I'll call, a feature vector, which I'll call x vector, which will be like x1 through, I think I use the subscript capital N. So it's an n-dimensional feature space. And so each one of those we can view as an input, which is kind of like a dendrite. So each one of those you can imagine has its own dendrite that will be summed in the body of the neuron. So I'm going to draw these x1, x2, x3, all the way down to xn. And then I am going to run each one of them through a gain so that the thing on the other side of it is just the gain multiplied by the signal. So I'm going to refer to these as a weight, W-E-I-G-H-T. So I'm weighting the inputs. And so these weights I'll be able to change, just like you can change how sensitive different synapses are to different neurotransmitters. And so each one of these has got its own individual weight. X3. And repeat that through the whole feature vector. And then I might add to it one more thing that's kind of like an auxiliary feature bias. And this bias is kind of a free parameter where I don't really need to weight. So I'm not going to add a, well, let me do this. What I'm going to do is I'm going to make this a 1. And then I'm going to run it through a weight that I'll call B, which is my bias. And we'll see why that bias matters here in a bit, so that this thing comes out to be B. So these are kind of like features. So these are my features. Well, I guess the bias doesn't count. So these are my features from pre-synaptic neurons. So you can think of each one of them existing at a dendrite. And this somehow represents in kind of an internal sensitivity. So, and we'll see what I mean by that. Okay, so we take all of those in, and just like in the chemical case, we're just going to sum all of their actions together. So I'm just going to draw a sum junction and connect everybody to that. So that the output here is just going to be, and I'll call the output X bar, which is just going to be the sum of all of the features plus the bias term. So maybe I'm going to move this over so I can put the bias in front of it so that it's clear the bias isn't in the sum. X bar is the bias term plus this guy. And that's what is going on this signal here. And then the last thing I've put in here is a function that I'll call f of x bar. You could call a of x bar, which is fine too. And that produces my output y. And this function we call an activation function. And this basically you could view it as assigning membership. We'll see what I mean by that. More generally, it transforms X bar to desired output space. And so the analog here would be, this is kind of the weighted combination of synaptic potentials. And you can view the activation function here as modeling the threshold for an action potential. So it's a basic arithmetic model of a neuron. And we can then see what we can do with this. So any questions about this basic model before we start interpreting it? See, make sure online, just the same questions as before. Okay, pretty clear. All right, so if you take in any sort of more sophisticated stats courses, you may have run into generalized linear modeling. And so if you haven't taken a more sophisticated stats course, you probably took a stats course once upon a time where you learned about linear models, which were just regression functions. And so you learned how to fit lines to data. And the basic idea there is it could give you inputs, I could give you outputs, and I could get a best fit line that would go through there. And that would represent the hypothetical linear relationship between whatever your dependent and independent variables were. Generalized linear models do the same thing, but before they fit a line, they transform the input space in a way that that might be better suited for linear regression. So, for example, if I wanted to figure out the different, like if I had two classes, like I wanted to figure out what's the difference between, what is a good predictor of whether someone wears blue one day or red another day. Those are my two classes, blue or red. I can label them 0 and 1. And I can look at all these features out there, like their height, their gender, all these different things. All of these things can come in, and those would be like my independent variables. But my output variable would be just two things, 0 or 1. Well, that by itself doesn't sound like a regression problem, because a regression problem means I can get a continuous input, continuous independent variable, and a continuous dependent variable. So what general linear models or generalized linear models do is they say, maybe we can transform the 0 to 1s in kind of you run them through a function so that instead of going from 0 to 1, they go from like negative infinity to infinity. So we're effectively mapping the 0 to negative infinity and the 1 to infinity. And then we're trying to do a regression problem to fit those things. And so it's kind of unbounded. And so it maybe is better suited for that. And so you create a link function, which turns the response variables you care about into these hypothetical response variables that are more likely to have a linear relationship to your input data. And that's exactly what's going on with this activation function. So this thing, if you're familiar, this is related to the link function in generalized linear modeling or models, GLM. So regression problems. So this is just kind of a generalized regression problem. And so what I mean by that, well, we'll see what I mean by that here in a second. Let's look at a couple of these link functions, or look at a couple of these, activation functions. So what are the types of activation functions that I might put in here? So activation functions. And so if I want to kind of model the most cartoon version of a neuron, which is kind of like for binary classification, I can imagine the output is either on or off. So I can use a threshold activation, which is otherwise known as a Heaviside step function. And in that case, my f of x bar is just going to be equal to, maybe I'll make this a little smaller, 1 if x bar is greater than or equal to 0, and 0 otherwise. I'll say 0 if x bar is less than 0. And so this is particularly good for binary classification. So in other words, what I mean by that is if I've got a bunch of features, so I can know that every person who wears a blue shirt, I've got all of their data. And each one of those I list as a feature. Everybody who wears a red shirt, I've got all of their data, and I list all those as a feature. And I would like to build a predictor where if you give me someone's features, I will predict whether they will wear a blue shirt or a red shirt. And that would fit this activation function. So in other words, if it, I'm not saying it's possible, but assuming it is possible, we could come up with a set of weights for this neural network here, or for this neuron, so that those weights would properly scale up or scale down features that are predictive. So it might be that if they're wearing dark colored shoes, that is a that typically is associated with wearing a blue shirt. And if they're wearing light colored shoes, that's typically associated with wearing a red shirt. So the color of their shoes might be this feature. And if the output here, if one is a blue shirt, then this weight, I might make a high positive weight because the color of their shoes tends to correlate very well with the color of their shirt. So in theory, I could choose these weights so that the X bar here will be a large number if they're likely to choose a blue shirt, and will be a small number if they're likely to choose a red shirt. And then I can then use this threshold to say if they are above, if it's very positive, I'm going to assign them a likely blue. If it's very negative, or if it's zero, then I'm going to assign them a likely red. So this is what we refer to as a binary classification problem. So that's kind of the simplest case here. These threshold functions don't tend to be used very often in practical neural networks where we have chains of these things together, because what we're going to find is that the way that they tune the weights of these neural networks is by using gradients. So they try to detect the how good a neural network is doing in terms of the gradients of their weights. And so I could say, what's the performance of this network in terms of this weight? And if my activation functions are discontinuous like this, then they don't give me a lot of gradient information. In other words, if I change the weight a little bit on the upstream side here, if I change these weights, it's not going to change the output very much, because so it kind of gets hidden by the threshold function. So typically they like to choose continuous activation functions, because then small changes of these weights actually make changes on the outputs. So if you're looking at the output, you can kind of say that, oh, look, we made a small positive change here, which created a positive change here. Let's keep making a positive change here so that we end up getting an output more and more like we want. So because these are discontinuous, these are not commonly used, at least in the kind of main body of a giant neural network. They might be used at the output of a neural network, but if you're combining lots of neurons together, the ones in the middle typically won't use this because they have this gradient problem. So there are other threshold or other functions we could use. So I could go the simpler one, linear. And that is the kind of the simplest activation function, where it doesn't even look like an activation function. It's just, in this case, I just drew it as an identity function. So in this case, this is more for linear regression. I hope you can see that if I make my activation function just equal to X bar, then the output is just the sum of the weighted inputs. That's exactly a linear regression. So this here turns a single neuron into a linear regression problem. So it's just a visualization of linear regression. That's all it is. So I can kind of combine the two of these and create what's called a rectified linear unit. So a rectified linear activation function. Which is sometimes called a ReLU or an ReLU, which is the rectified linear unit, which is a very popular nowadays. And it is just linear where you clip off the negative. So it's just, it looks a little bit like the unit step. It'll be X bar if X bar is greater than or equal to 0, and it'll be 0 if X bar is less than 0. And so sometimes this is called a hinge function or a ramp function. So it's just kind of another common thing where it will end up being very useful inside the kind of hidden layers of these things. And then if we do want something that looks more like the Heaviside step function, but it doesn't have this ugly discontinuity, then we use sigmoidal functions. Sigmoid is a term that just means S-shaped. Remember, boid means like bird-like, bird-oid. And so sigmoid is that moid is like, and sig comes from a Latin for kind of S. And so sigmoid just means S-shaped. And there are two common kind of sigmoidal functions. The classic sigmoidal function is the logistic. And the logistic just looks like f of x bar is equal to 1 divided by 1 plus the exponential of minus x bar. And this has a shape which is going to look similar to the Heaviside. But so if x bar is very, very large, then this exponential here is going to be 0, and this will just map to 1. And if x bar is very, very small, so if x bar is equal to 0, then this will be 1/2. And if x bar is very, very small, then this negative will make it very, very large, which will make the exponential very, very large, which will make this 0. So this logistic maps from 0 to 1, similar to the Heaviside, but it has kind of a smoother case here where it kind of like ramps up and then comes here. So it does end up having an asymptote at 1. And then when x bar is equal to 1, it's at 1/2. And it goes to negative infinity. But as it gets closer and closer to negative infinity, it gets closer and closer to 0. So it never actually reaches 0, just closer and closer to 0. And the other, and this is the classical sigmoid. So if somebody in a paper writes, we used a sigmoidal activation function, and they don't tell you what they mean, if they used it correctly, this is what they mean. If they used it incorrectly, they mean the next thing, which is also sometimes called a sigmoid, but this is the classical sigmoid. So by default, if they don't tell you what they mean when they say sigmoid, they mean this thing. The other sigmoid that people like to use is a hyperbolic tangent. Otherwise known as a tanh. And the hyperbolic tangent is just f of x bar is equal to, and it's going to share some features with the sigmoid, 1 minus exponential, and I'll do minus 2x bar over 1 plus exponential minus 2x bar. And that again is what we would call the tanh function. And so what goes on with the hyperbolic tangent? Well, if you think about it, if the exponential here is, let's say x bar is giant. If x bar is giant, then both of these exponentials will go to 0, and this will just be equal to 1. If x bar is equal to 0, then this is going to be 1, and so the numerator here is going to be 0, so this will be 0. If x bar is very, very negative, like negative infinity, then this will be very large, as will this. Their magnitudes will be the same. They'll kill these ones, and this will go to -1. So this is a sigmoidal activation function that goes, instead of going from 0 to 1, it goes from -1 to 1. And it has a slightly different shape. So if I were to, I'm going to try to draw in the corner here, 1 and a -1, and this kind of going from -1 up to 1, where it kind of hits the middle, it hits at x bar is equal to 0, it's hits 0. So I don't know if that's a great illustration, but you can look up a tanh function or plot it on your calculators or whatever. So, so this hyperbolic tangent, its range is from -1 to 1. The logistic, its range is from 0 to 1. Both of these are S-shaped, and both are sometimes called sigmoidal, although the classic sigmoid is the logistic. All right, so those are common activation functions that you're going to see used across a bunch of neural networks. All right, so So any questions about these formulations here? I see online. There's a question online, is this similar to logistic regression? And yeah, logistic regression is an example of generalized linear modeling. So when I, back here, when I said this is related to the link function, GLM, I could have said example, logistic regression. So logistic regression is an example where your link function ties a continuous output to an output which actually has that logistic. So this logistic maps a continuous output from negative infinity to a response variable that goes from 0 to 1. So when you use an activation function, which is the logistic, then this does very much look like logistic regression because you're saying that I have built a model that produces a 0 to 1 output using an input here, where if I were to look at this level before the activation function, I would get something that would look more like standard linear regression. But when I look at it here, it's logistic regression. So yes, you can. That's what logistic regression is, an example of generalized linear modeling. So there are a bunch of other types of link functions that lead to other types of regression. And so if you're familiar with logistic regression, that's probably because you're familiar with data sets that are 0, 1. But if you look at a lot of other data sets, then there's a bunch of other regression types that just pick different link functions, which are equivalent to picking different activation functions in neural networks. Yeah.
Is it softmax is also X-shaped?
So softmax, that's getting a little more advanced. So right now I'm only talking about a single neuron with a single output. And so when we talk about multi-layer perceptrons, which we'll start, I think, talking about tomorrow or Tuesday, then we actually have multiple outputs. And when you want to do a classification problem, when it's not a binary classification, when you want to choose among multiple classes, then there are different ways of dealing with that. And softmax, which is related to the Boltzmann distribution, provides a way of sort of assigning probabilities and then choosing among those probabilities among multiple outputs. So if in my example, I predicted red or blue shirts. Well, if I wanted to predict 10 different colors of shirts, then my features are going to give me different accumulations for each color of shirts. So it'll be this, this, if this is the red line, it might be some number. If this is the blue line, it'll be some number. If I imagine multiple of those neurons that all in another layer are being kind of pulled together, then I would say, well, I can use this so-called softmax function, which again is like the Boltzmann distribution, to turn these numbers into a probability distribution that sums to 1. And then that probability distribution could be viewed as an output, like a posterior probability, or I could choose like the maximum of those. So that's like, soft argmax. And then that would end up saying, okay, that gives me a decision like red shirts or something like that. So that's getting a little more advanced, but it's certainly related to all of this. All right. So any other questions about that? I think this is a good spot to stop. And so what basically we're going to do starting next time is we're going to take this and we're actually going to see mathematically how this, so what is the relationship between the feature vector and the weight vector? And there's a nice geometric relationship where we're basically going to plot the feature space. So that's the X space. And in that X space, we'll be able to then draw a line through the X space that's encoded by the weight space that cuts the feature space into 2 sections. And one of those sections will be the blue shirts and the other sections will be the red shirts. In other words, one of the sections will be the 0 output, the other section will be the 1 output. And our goal in training the neural network is to find the right line. So this is sort of like the slope of that line. And so we need to find the right slope of that line. And that's, maps to linear regression. I got a bunch of points and I know there's a line that fits through there and I need to find the intercept. Well, this bias is the intercept and the slope. Well, this is like for a simple linear regression problem, I only have one of these and a bias. and this is the intercept, and this is the slope. And so we know how to do that for a single variable. This allows us to do it for multiple variables, and geometrically, we'll see it's just going to separate a multi-dimensional space into two regions for our binary classification. And then we'll talk about, well, what if it's not linearly separable? What do we do about that? And then how do we work on more complicated problems? And how are those inspired by brain structures? So that's where we're going. But meanwhile, that's the end. That's all I've got for you today. There's a muddiest point due on Sunday. Remember, I made the mini project 4. I think I allowed you to turn it in until Sunday, but mini project 5, I think, is also due currently on Sunday. I think that's it. I think I've got all of the project groups in except for one, where there's one person out there who I've been emailing to try to get to, so I get you into a group. So if you're listening, you know who you are, respond to me so I can get you into a group and finally finalize all the groups. But otherwise, if you haven't heard from me and you submitted your group thing, your group's good and you can keep moving on. So that's all I got for you. You can get out of here. If you have any other questions, feel free to come up after, and we'll have a good weekend. See you next week. All right, any other questions online? If not, then I hope all of you have a good weekend, and we'll see you next week.